Dear viewers, this video is part 1 of the two-part series on the red blood cell in the Medicine for Anyone in the YouTube channel. Red blood cells. Red blood cells, also known as erythrocytes, are the most abundant cells of the blood. They are necessary for the delivery of oxygen to the tissues. The body produces 2.4 million red blood cells per second. Hemoglobin, situated inside the RBCs, carries oxygen from the lungs to the tissues. In some animals, including many invertebrates, hemoglobin circulates as free protein in the circulatory fluids and is not enclosed in RBCs. When it is free in human plasma, about 3% of it leaks through the capillary membrane into the tissue spaces or through the glomerular membrane of the kidney into the glomerular filtrate each time the blood passes through the capillaries. Therefore, hemoglobin must remain inside the RBCs to perform its functions in humans effectively. The RBCs have other functions besides transport of hemoglobin. For example, they contain a large quantity of carbonic anhydrase, an enzyme that catalyzes the reversible reaction between carbon dioxide and water to form carbonic acid, increasing the rate of this reaction several thousandfold. The rapidity of this reaction makes it possible for the water of the blood to transport enormous quantities of carbon dioxide in the form of bicarbonate ion from the tissues to the lungs, where it is reconverted to carbon dioxide and expelled into the atmosphere as a body waste product. The hemoglobin in the cells is an excellent acid-base buffer, as is true of most proteins. So, the RBCs are responsible for most of the acid-base buffering power of whole blood. Dear listeners, by the way, this educational video is made possible by Prescribe-On, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribeon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. This picture shows a red blood cell. Red blood cell is a round biconcave disc-shaped structure, 7.8 micrometer in diameter, a variable thickness of 1 to 2.5 micrometer, with a volume of 90 cubic micrometers, containing hemoglobin, and has a lifespan of 120 days in the blood. Shape and size of red blood cells. See the figure earlier shown, red blood cell. Normal RBCs shown in the figure. These are biconcave discs having a mean diameter of about 7.8 micrometers and a thickness of 2.5 micrometers at the thickest point and 1 micrometer or less in the center. The average volume of the RBC is 90 to 95 cubic micrometers. The shapes of RBCs can change remarkably as the cells squeeze through capillaries. Actually, the RBC resembles a bag that can be deformed into almost any shape. Furthermore, because the normal cell has a great excess of cell membrane for the quantity of material inside, deformation does not stretch the membrane greatly and consequently does not rupture the cell, as would be the case with many other cells. Concentration of red cells in the blood. The average number of RBCs per cubic millimeter is 5.2 million. In healthy women, it is 4.7 million. Persons living at high altitudes have greater numbers of RBCs. Quantity of hemoglobin in the cells. RBCs can concentrate hemoglobin in the red blood cell fluid up to about 34 gram per 100 ml of cells. When hemoglobin formation is deficient, the percentage of hemoglobin in the cells may fall considerably below this value, and the volume of the RBC may also decrease because of decreased hemoglobin level to fill the cell. When the hematocrit, that is the percentage of blood that is comprised of cells, normally 40 to 40%, and the quantity of hemoglobin in each respective cells are normal, the whole blood of men contains average of 15 gram hemoglobin per 100 ml. For women, it contains an average of 14 gram hemoglobin per 100 ml. Each gram of hemoglobin can combine with 1.34 ml of oxygen if the hemoglobin is 100% saturated. Therefore, in the average man, a maximum of about 20 milliliters of oxygen can be carried in combination with hemoglobin in each 100 milliliters of blood, and in women, 19 milliliters of oxygen can be carried. Production of red blood cells. Areas of the body that produce red blood cells. In the early weeks of embryonic life, primitive nucleated RBCs are produced in the yolk sac. Note: yolk sac is a structure that develops inside the uterus during early pregnancy. It provides the embryo with nourishment. It helps circulate carbon dioxide and oxygen between the uterus and the embryo. The yolk sac also produces cells that turn into important structures such as the umbilical cord, blood cells, and reproductive organs. During the middle trimester of gestation, the liver is the main organ for RBC production, but reasonable numbers are also produced in the spleen and lymph nodes. Note: spleen is a fist-sized organ in the upper left side of the abdomen, next to the stomach and behind the left lower ribs. It's an important part of the immune system, but one can survive without it. This is because the liver can take over many of the spleen's functions. Then, during the last month or so of gestation and after birth, RBCs are produced exclusively in the bone marrow. Note: bone marrow is the spongy tissue inside the bones. It contains stem cells. The stem cells are mother cells that can develop into red blood cells, white blood cells, and platelets that help with blood clotting. The marrow of essentially all bones produces RBCs until a person is about 5 years old. The marrow of the long bones becomes fatty and produces no more RBCs after about the age of 20. But the proximal, that is nearer to the body, portions of the humerus and tibia continue to produce RBCs even after 20 years. Beyond 20 years of age, most RBCs continue to be produced in the marrow of the membranous bones such as the vertebrae, sternum, ribs, and ilia. Even in these bones, the marrow becomes less productive as age increases. Genesis of blood cells. Multipotential hematopoietic stem cells, growth inducers, and differentiation inducers. The blood cells begin their lives in the bone marrow from a single type of cell called the multipotential hematopoietic stem cell, from which all the cells of the circulating blood are eventually derived. With the clinical use of hematopoietic stem cells, tens of thousands of lives are saved each year. Stem cells produce hundreds of billions of blood cells daily from a stem cell pool that is estimated to be only 100,000. These cells produce 4,900 trillion red cells in a human lifetime at a 70-year estimate. We are still in the process of learning how stem cells do this and how they persist for many decades despite the production demands. This figure shows how multipotent stem cells maintain their stem cell pools and at the same time differentiate or mature into erythrocytes, granulocytes, which are neutrophils, eosinophils, and basophils, monocytes, and megakaryocytes, as well as T lymphocytes and B lymphocytes. Formation of blood cells in bone marrow. See the figure earlier shown. This shows the successive divisions of the multipotential cells to form the different circulating blood cells. As these cells reproduce, a small portion of them remains exactly like the original multipotential cells and is retained in the bone marrow to maintain their supply. They are called stem cells, which are having a character of self-renewal, although their numbers diminish with age. Most of the reproduced cells, however, also differentiate to form other cell types, shown at the right side of the same figure. The intermediate stage cells are very much like the multipotential stem cells, even though they have already become committed to a particular line of cells. These are called committed stem cells. A committed stem cell that produces erythrocytes is called a colony forming unit, erythrocyte, and the abbreviation CFUE is used to designate this type of stem cell. Likewise, colony forming units that form granulocytes and monocytes have designation CFUGM and so forth. Growth and reproduction of the different stem cells are controlled by multiple proteins called growth inducers. At least 4 major growth inducers have been described, each having different characteristics. One of these, interleukin 3, promotes growth and reproduction of virtually all the different types of committed stem cells. Others induce growth only specific types of cells. The growth inducers promote growth but not differentiation of the cells. Differentiation inducers produce differentiation into particular type of cells. Each of these differentiation inducers causes one type of committed stem cell to differentiate one or more steps toward a final adult blood cell. Formation of the growth inducers and differentiation inducers is controlled by factors outside the bone marrow. For example, in the case of RBCs, exposure of the blood to a low oxygen level for a long time causes growth induction, differentiation, and production of greatly increased numbers of RBCs. In the case of some of the white blood cells, infectious diseases cause growth, differentiation, and eventual formation of specific types of white blood cells that are needed to combat each infection. This figure shows the various stages of maturation of RBCs. The differentiated mother cell is the proerythroblast, which goes through the following stages: basophil erythroblast, polychromatophil erythroblast, in which the first signs of formation of hemoglobin is recognizable, then the orthochromatic erythroblast, then the reticulocyte, where the nucleus condenses, become inactivated and is extruded from the cell. The reticulocyte enters the circulation and becomes a mature RBC, which lives for 120 days in the circulation. Stages of differentiation of red blood cells. Figure stages of maturation of RBC shown earlier. Please refer to it. The first cell that can be identified as belonging to the RBC series is the proerythroblast, shown at the starting point in figure shown above. Under appropriate stimulation, large numbers of these cells are formed from the CFUE stem cells. Once the proerythroblast has been formed, it divides multiple times, eventually forming many mature RBCs. The first generation cells are called basophil erythroblasts because they stain with basic dyes. Hemoglobin first appears in polychromatophil erythroblasts. In the succeeding generations, the cells become filled with hemoglobin to a concentration of about 34%. The nucleus condenses to a small size and its final remnant is absorbed or extruded from the cell. At the same time, the endoplasmic reticulum is also reabsorbed. Note: other vertebrates such as fish, reptiles, and birds have red cells that contain nuclei that are inactive. Losing the nucleus, as in humans, enables the red blood cell to contain more oxygen carrying hemoglobin, thus enabling more oxygen to be transported in the blood and boosting metabolism. The cell at this stage is called a reticulocyte because it still contains a small amount of basophilic material consisting of remnants of the Golgi apparatus, mitochondria, and a few other cytoplasmic organelles. During this reticulocyte stage, the cells pass from the bone marrow into the blood capillaries by diapedesis, that is, squeezing through the pores of the capillary membrane. The remaining basophil material in the reticulocyte normally disappears within 1 to 2 days, and the cell is then a mature erythrocyte. The mean transit time from proerythroblast to the emergence of the reticulocyte into the circulation is approximately 10 to 14 days. Because of the short life of the reticulocytes, their concentration among all the RBCs is normally slightly less than 1%. So, dear viewers, this is the end of part 1 of the two-part series on the red blood cells in the Medicine for Anyone in the YouTube channel. Thank you for watching the video.